Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The 2017 Nobel lectures delivered by Jeffrey Hall, Michael Rosbash, and Michael Warren Young represent a capstone moment in modern biology, a formal chronicle detailing the decades-long intellectual and experimental journey into the heart of life's intrinsic rhythms. This essay undertakes a rigorous evaluation of the claims, evidence, and conceptual frameworks presented within this collective lecture, treating it as a singular, cohesive scientific testament. The analysis will proceed through the sequential logic of the discoveries themselves, as narrated by the laureates, to assess the internal coherence and explanatory power of their account. It is an examination of a story told by its authors, a story of how a fundamental property of physiology, the circadian clock, was resolved from a behavioural enigma into a mechanism of exquisite molecular precision. The inquiry here focuses exclusively on the content of the lecture itself, understanding it as a primary document that both summarises and canonises a revolutionary body of work for the annals of science. Section 1, establishing a genetic locus for a complex behaviour. The lecture's narrative foundation rests upon a powerful initial claim that a complex, organism-wide behaviour, the daily cycle of activity and rest, is governed by discrete, discoverable genetic elements. The laureates ground their entire subsequent exposition in the foundational work of Seymour Benser and his student Ronald Konopka, whose forward genetics screen in Drosophila melanogaster yielded the first cock mutants. The lecture presents this 1971 discovery as the conceptual key that unlocked the entire field. The evaluation of this claim is straightforward. Its strength lies in its profound simplicity and empirical verifiability. The laureates detail the isolation of three distinct alleles of a single gene, which they designated period. One allele, period short, produced flies with a roughly 19-hour cycle. Another, period long, resulted in a 29-hour cycle. A third, period null, produced rhythmic flies. The lecture's argument is that this triad of phenotypes unequivocally demonstrated a direct, causal link between a specific genetic locus and the temporal regulation of behaviour. The evidence presented is the behavioural data itself, the elegant eclosion and locomotor activity, rhythms that could be precisely quantified. The intellectual weight of this claim, as framed in the lecture, is immense. It transformed a problem of physiology into one of molecular genetics. The laureates are careful to position this early work as providing the essential toolkit and the central question for their own research: what is the molecular identity of the period gene, and how does it function to keep time? The lecture structure implies that without this initial, behaviour-centric genetic discovery, the molecular deconstruction that followed would have been impossible, a claim well supported by the history of the field. The work of Benser and Konopka provided the essential entry point, an identifiable genetic address for an abstract biological process. Section 2, the molecular cloning and initial characterization of the period locus following the establishment of the period gene's existence. The next major section of the laureate's narrative addresses the monumental task of its molecular identification. Both the Hall and Rosbash group and the Young group independently embarked on this challenge. The lecture frames this effort as a heroic feat of early molecular biology, a chromosome walk spanning vast, uncharted regions of the Drosophila genome. The claim here is one of technical perseverance leading to a breakthrough. They detail the painstaking process of using overlapping DNA clones to move incrementally along the chromosome toward the genetically mapped location of the period gene. The evidence for their eventual success is the identification of a specific DNA segment that, when transferred into a rhythmic, period null mutant flies, restored a normal 24-hour rhythm. This gene rescue experiment, as presented, serves as the definitive proof that they had cloned the correct gene. The lecture then moves to evaluate the initial findings from sequencing the period gene. A subtle but crucial claim is made here regarding the nature of scientific discovery itself. The amino acid sequence of the resulting period protein, which they call per offered few immediate clues to its function. It lacked homology to any known enzymes or structural proteins of the time. However, it did contain a motif they identified as the PAS domain, a sequence later found in other proteins involved in environmental sensing. The lecture presents this finding with a touch of historical irony. The PAS domain was a hint, a clue to a future understanding of protein-protein interactions, yet at the moment of discovery, its full significance was opaque. The evaluation of this part of the narrative reveals a core theme. Discovery is an iterative process of finding pieces of a puzzle whose final picture is unknown. The cloning of the period gene was a success, but it yielded a new, more complex mystery regarding the biochemical function of its protein product. Section 3. The central postulate of a transcriptional negative feedback loop, perhaps the most conceptually elegant claim in the entire Nobel lecture, is the proposition of the autoregulatory negative feedback loop as the core timekeeping mechanism. This section of the narrative is largely driven by Michael Rosbash's account of his lab's pivotal discovery. The central assertion is that the period gene product, the protein, functions to inhibit the transcription of its own gene. The evidence presented for this hypothesis is a series of measurements of the abundance of period messenger RNA and protein over a 24-hour cycle. The lecture details the finding that period messenger RNA levels peak in the early evening, while the accumulation of protein is delayed, peaking several hours later in the middle of the night. This temporal lag is presented as the critical observation. Rosbash explains the logical inference. The delay between the peak of the transcript and the peak of the protein suggests a post-transcriptional or post-translational regulatory step. More importantly, as protein levels rise, period messenger RNA levels begin to fall. This inverse relationship is the cornerstone of the negative feedback model. The lecture claims that the accumulating protein, after a delay, enters the cell nucleus and acts as a repressor, shutting down the activity of its own gene. This shutdown then leads to a decline in protein levels, which in turn relieves the repression, allowing the transcription of the period gene to begin anew, thus starting the next cycle. The power of this claim lies in its self-contained logic. It provides a complete, cyclical explanation for a biological oscillation. It is a beautiful and parsimonious model that accounts for the rhythmic nature of the clock with a minimal set of components, a gene and its inhibitory protein product. The evaluation of this claim within the lecture's context shows it to be the intellectual centerpiece of their discovery, a framework upon which all subsequent findings would be built. Section 4. The discovery of timeless as the obligate partner. While the negative feedback loop provided a powerful conceptual model, it contained unresolved questions, chief among them the source of the delay between transcription and repression. This is where Michael Young's contributions, as detailed in his portion of the lecture, become essential. Young's lab conducted a parallel genetic screen for clock mutants and identified a second crucial gene, which they named timeless. The central claim presented in this section is that the TIM protein, the product of the timeless gene, is an indispensable partner to the protein. The lecture details the evidence for this partnership through a series of genetic and biochemical experiments. Young explains that in timeless mutant flies, the protein is produced, but it remains highly unstable and is rapidly degraded in the cytoplasm. It fails to accumulate to high levels and, crucially, fails to enter the nucleus. The claim is that TIM acts as a chaperone or stabilizer for BA. The evidence becomes more direct with the demonstration that the two proteins physically associate. The lecture posits, at PA and TIM form a heterodimeric complex, a stable unit composed of one molecule of each protein. It is this PA-TIM complex, they claim, that is capable of accumulating in the cytoplasm and subsequently translocating into the nucleus to perform its repressive function. This discovery elegantly solved two problems at once. First, it provided a mechanism for stabilizing the otherwise labile protein. Second, the time required for TIM protein to accumulate and bind with PA provided a plausible explanation for the hours-long delay observed by Rosbash's group, the very delay that is critical for the 24-hour pacing of the clock. The evaluation of this claim shows how it refined and strengthened the original negative feedback model, transforming it from a simple loop into a more complex, multi-component molecular machine. Section 5 Phosphorylation as the kinetic pacemaker. The narrative of the lecture continues to add layers of regulatory complexity, moving from the identity of the core components to the mechanisms that control their timing. The next major claim, again emerging from Young's work, is that protein phosphorylation acts as the master regulator of the clock speed. This section focuses on the discovery of yet another gene, double time, which encodes an enzyme called casein kinase epsilon. The lecture asserts that this enzyme is responsible for the precise 24-hour period of the clock by controlling the stability of the protein. The evidence presented is compelling. Mutations in the double time gene have a dramatic effect on the period length, mirroring the effects of the original period gene mutations. One allele creates a short period clock, while another leads to a long period clock. The biochemical claim is that the double-time kinase phosphorylates the protein at specific sites. This phosphorylation is presented as a molecular switch. When PER is alone in the cytoplasm, phosphorylation by double-time targets it for rapid degradation. This prevents premature accumulation and nuclear entry. However, the lecture claims that when performs a complex with TRM, it becomes protected from the full effects of double-time immediate phosphorylation. This protection allows the PERTIM complex to accumulate, providing the necessary delay before it enters the nucleus. Furthermore, once inside the nucleus, additional phosphorylation events are proposed to be critical for the eventual disassembly of the repressive complex and the degradation of PER and TRM, which is necessary to start the next cycle. The evaluation of this claim is that it provides a sophisticated kinetic control mechanism. The genius of this system, as described, are the constant push and pull between the stabilizing influence of TRM and the destabilizing influence of double-time phosphorylation creates a robust, tunable delay, turning a simple on-off switch into a precise molecular chrono. Section 6 Completing the circuit with positive elements clock and cycle. A negative feedback loop requires an initial on signal to function. The laureate's lecture addresses this in a section dedicated to closing the loop. The core claim here is the identification of the transcriptional activators at turn on the period and timeless genes in the first place. The work, primarily attributed in the lecture to Joseph Takahashi's discovery of the clock gene in mice and its subsequent characterization in flies by the laureate's groups, identified two key proteins, clock and cycle. These two proteins are presented as forming another heterodimeric complex, this time a transcription factor. The lecture claims that the clock cycle complex is the positive element of the clockwork. It binds to specific DNA sequences known as E-boxes, which are located in the promoter regions of the period and timeless genes, as well as other clock-controlled genes. This binding actively drives the transcription of period and timeless, initiating the entire cycle. The negative feedback loop is thus completed. Clock and cycle turn the genes on, leading to the production of PER and TRM proteins. The PERTIM complex then accumulates, enters the nucleus, and physically interacts with the clock cycle complex, inhibiting its ability to activate transcription. This turns the genes off. As PER and TRM are eventually degraded, the inhibition is relieved, allowing clock and cycle to begin their work again. The evaluation of this part of the lecture demonstrates the completeness of the model. It now contains both the positive clock cycle and negative PERTIM elements required for a self-sustaining oscillation. The lecture presents this fully realized transcription translation feedback loop as a fundamental gear of the circadian clock, a claim that has become dogmatic in the field, a testament to its explanatory power as self-evidence marshaled in its Section 7 The Conservation of the Clockwork across phyla. The final major section of the laureate's collective lecture extends their claims beyond the fruit fly, asserting the profound evolutionary conservation of the core timekeeping mechanism. The argument presented is that the molecular machinery they elucidated in Drosophila serves as a blueprint for the circadian clocks found in a vast array of organisms, including mammals and humans. They provide evidence by highlighting the discovery of mammalian orthologs for the key fly genes. The mammalian genome contains multiple period genes, such as period 1, period and period 2, and cryptochrome genes, cryptochrome 1 and cryptochrome 2, which serve a role analogous to that of thymus in flies. The mammalian orthologs of clock and cycle, the latter known as BMAL1 in mammals, also function as the core positive activators. The lecture claims that these mammalian proteins interact in a transcription-translation feedback loop that is strikingly similar in its logic and architecture to the one found in Drosophila. The PO and CRY proteins form complexes that enter the nucleus to inhibit the transcriptional activity of the clock-1 complex. The role of casein kinase iron phosphorylating the PO proteins to control timing is also conserved. The evaluation of this claim is that it elevates the significance of their work from a specialized finding in insect biology to a universal principle of life. The lecture effectively argues that their painstaking dissection of the fly clock provided the direct intellectual and genetic tools needed to rapidly uncover the basis of human circadian rhythms, with profound implications for health, disease, and medicine. This claim of conservation solidifies the Drosophila model as one of the most powerful and productive in the history of biology. In conclusion, the 2017 Nobel lecture on circadian rhythms by Hall, Rosbash, and Young is a masterclass in scientific exposition. It constructs a compelling and logically airtight narrative that guides the audience from a simple behavioral observation to the intricate workings of a molecular machine. The claims presented at each stage, from the genetic basis of rhythmicity to the roles of protein partnership and phosphorylation in setting the clock's pace, are substantiated with specific, elegant experimental evidence. The evaluation of the lecture's internal arguments reveals a story of parallel and collaborative discovery, where each new finding resolved a previous puzzle while simultaneously adding a new layer of beautiful complexity to the emerging model. The final claim of evolutionary conservation serves as a powerful testament to the fundamental nature of their discoveries. The lecture functions as more than a summary of data. It is the definitive, first-person account of a scientific revolution and document that not only explains how the clock works but also demonstrates the process of scientific inquiry at its most persistent, insightful, and ultimately triumphant. It codifies a paradigm, presenting a complete and satisfying explanation for one of life's most ubiquitous and mysterious phenomena.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.